My name is Kathy Hernandez. I'm in group 1. I'm doing member 4 topic. We're looking at techniques in cell biology, specifically flow cytometry. Biologists use flow cytometry in order to identify and quantify specific cells with set properties. So, flow cytometry provides automated measurements of cells. This is beneficial because it provides rapid measurements based on specific properties such as cell size. This can be also combined with a technique called fluorescent activated cell sorting. So, this produces a purified population that can be further observed and studied. Flow cytometry has a list of methods, but some of the most common methods for flow cytometry begin with a mixture of cells. This mixture of cells is called cells in suspension. They are then treated with different dyes. Some can be fluorescent antibodies or antigens, and then some can also be dyed with a purple dye or a DNA dye to be able to look at the concentration of DNA. So the cells are then placed in the flow cytometer where the cells flow down in a single file line. These single cells are then hit with a laser where the fluorescent light intensity is measured by a fluorescence detector and the scatter light is used to measure cell shape and size. So in flow cytometry, we begin by staining these cells with one or more fluorescent dyes, each dye correlating with a specific set of properties in identifying that cell that we're looking at. So some dyes can include a purple dye, which is a DNA dye that can be used to observe the concentration of DNA in individual cells. So the next type of dye is a fluorescent green dye, which can bind to specific extracellular proteins on the surface of cells, and we can observe the presence of those specific proteins. So, some of the main components of flow cytometry that we will look at are laser beams, which assess the fluorescence of the cells or the quantity. So, this would be looking at the light intensity of the fluorescence, and then the scatter light provides information on the shape and size of the cells. We would be looking at the side scatter for this section, and then antigens are used in order to observe the presence of membrane glycoprotein. So we can use different antigens in order to observe the different or specific membrane glycoproteins. This figure comes from an article titled KCTD15 Protein Expression in Peripheral Blood and Acute Myeloid Leukemia. In this research article, the researchers are looking at leukocytes, which are major cellular components in inflammatory and immune response. Leukocytes are white blood cells that derive from hematopoietic stem cells in bone marrow. So these mature white blood cells include lymphocytes, monocytes, and granulocytes. These cells display similarities and differences in expression of specific cell surface receptors, such as CD45, which is a common pan-leukocyte antigen. So in this figure, 1C, CD45 and the side scatter color plot shows the identification of the lymphocytes, the monocytes, and the granulocytes based on their CD45 intensity of expression and light side scatter. So the light side scatter measures the complexity and granularity of the cell, which is the inner part of the cell, and the CD45 intensity or expression helps with identifying the specific types of cells, which are the lymphocytes in this area in the blue, and then the monocytes in the orange, and the granulocytes in the red. So, how we're talking about how the CD45 antigen binds to a specific membrane glycoprotein expressed in most hematopoietic cells. So, furthermore, there is another figure that used an additional antigen to further identify these three specific leukocytes. So, CD14 and CD33 were then further used to identify the monocyte subsets, and then CD66B was used for the clustering granulocytes. My question is, what is or are the limitations when using a flow cytometry approach? These are my references. Number one was used in the discussion of the flow cytometry methods from the Becker's World of the Cell. And the second reference is used for the figure 1C in the KCTD15 protein expression in peripheral blood and acute myeloid leukemia article.